Right, so welcome. Apologies for the problem. It's a pleasure to introduce Alessia Annibale from King's College in London. She's been there for a while. She holds a degree from La Sapienza in Rome with Andrea Cavagna and in the group of Giorgio Parisi on a topic on supersymmetry in spin glasses. But at the time created quite some furore. And then did her PhD with Peter Solich at King's College. And then she's been there ever since and is now a reader in disordered systems. And she's talking about gene regulatory networks viewed through statistical mechanics glasses.
Okay. Thank you very much, Tobias. And thanks a lot for the invitation. It has been a real pleasure to be here. And oops. It's on. Yeah, it's green. Okay. It has been a real pleasure to be here and interact with several of you. So in this talk, I will first give you a brief overview of my research interest, and then I will focus on one in gene regulatory networks, and I will present two different modeling approaches I've been using, one based on neural networks and the other based on bipartite graphs. So, okay, so these are my main research interests, more or less in chronological order. So as Tobias mentioned, I started several years ago in spin glasses with Andrea Cavagna and Giorgio Parisi. The main focus at the time was to compute the number of metastable states that this system typically exhibits, which can be visualized as the local minima of a rough landscape, as illustrated by this picture produced by Chiara Camarota. And we were also interested in quantifying the structure of these states, because these are implications on the dynamics of the system. Then I moved to consider dynamics in non-equilibrium settings with a main focus on fluctuation dissipation violation with Peter Solich. And next, I initiated a new research line in complex networks, mainly with Tom Cullen and other collaborators as well. And a central question at the time was a good definition of null models. And this is because if I observe a network and observe some features, one of the first things I want to know is whether these are special or just normal, okay? And crucially, the answer to this question depends on our model of normal. And I was also interested in algorithms to generate random graph from these null models in an efficient way, but also unbiased way. Okay? Then I move on to consider network processes. So this is when you place stochastic variables on the nodes, and these variables interact through the links, which can be weighted. And the main aim is to understand how the network structure influences the process or affect the resilience of the process on top of it. And I've done this for different systems with different variables, spin, Kuramoto oscillator, and so on, with different applications in mind, okay? So I, for example, consider processes in biological complexity, where I modeled the immune systems or gene regulatory network, as well as epidemic spreading on network of contacts. And more recently in social systems, where, for example, I consider systems of voters or agents trading information. And more in general, I have an ongoing collaboration with Pierpaolo Vivo on trying to use network tools and statistical physics to make a legal system more quantitative, as this picture summarized. So I've always been very, very keen on cross-disciplinary collaboration, and this is a non-exhaustive list, but maybe I can mention one more in kinetic networks with the chemistry department. So kinetic networks are basically Markov models for stochastic trajectories, which in that context came out of molecular dynamic simulation, okay? And one of the central questions here is how to optimally coarse grain a system which has lots of microstates and transition between them into a reduced description with just a few effective states and effective transition between them. And okay, some of these projects have then inspired a new question on more theoretical aspects of information processing, on which I'm currently working on. And also they have sparked a keen interest on modeling hypergraph from both a point of view, null models, and also considering equilibrium, processing in equilibrium and non-equilibrium on hypergraph. So basically this was the chronological order, but these are all interconnected. So say the initial work laid the foundation for cross-disciplinary work, which then sparked new questions on more theoretical aspects and all the way back to more traditional disordered system. But this is it. And today I will focus on a theme which is maybe not the most recent, but has been really central in my research, which is biological complexity, and has generated lots of questions in this field where this institute has also a strong tradition. Okay. So I will focus on gene regulatory networks. The motivation, I think it all happened by chance. I just attended a beautiful seminar years ago by Rick Blank at King's on cell differentiation and cell reprogramming, and the topic really fascinated me. So cell differentiation, you probably all know about, is the process whereby embryonic stem cell divides and differentiates into pluripotent stem cell, which then divides and differentiates further until fully differentiated cells are reached. And this constitutes our body, okay? Neurons, blood, bones, and so on. And these cells contain exactly the same genes, but different cells express different genes, okay? And what regulates gene expression are transcription factors, which are either proteins or protein complexes, like groups of proteins, which bind to the DNA and decide which gene should express and which should be silent. Now, cell reprogramming is an experimental protocol pioneered by Takashi and Yamanaka in 2006. Whereby they introduce a cocktail of transcription factor, 4 transcription factor, which are now known as Yamanaka factor. They introduce this in a fully differentiated cell, and they see this to transition to a state of induced pluripotency, from which, in principle, one can differentiate them back into any desired cell type. And indeed, Yamanaka got the Nobel Prize for this important discovery in 2012. It takes about 10 days to reprogram a cell. And nowadays, these experiments are actually quite efficient. However, many cells are seen to take bad trajectories, so to speak, at later stages. So, for example, they can develop cancer later on, which, of course, limit the use of this experiment in vivo. So the big question here is how to control cell fate. Okay? And I thought perhaps before answering this question, we should understand how genes create cells in the first place. So when viewed through statistical mechanical glasses, cell types can be thought of as stochastic attractors of a complex gene dynamics, pretty much like memories are seen as the stochastic attractors of brain dynamics or neural networks. And then in this paradigm, within this paradigm, you could think of cell reprogramming as inducing transition from one attractor to another one, okay? So this is what inspired the first model In particular, we started with a preliminary observation, which is, well, if we knew all the chemicals, all the processes inside a cell, and if we wrote down equation for each single reaction, and then if we integrate out all the degrees of freedom except the genes, I would necessarily end up with a system of genes only, okay, integrated everything out, which effectively interact with each other, okay? So I could then say that the dynamics of a gene I will be influenced, will be determined by the other genes J, which interact with I through some couplings, okay? J. And I could take this to be positive if gene J promote expression of I, and negative if it inhibits. And then if we model in a simplistic way genes as Boolean variable, 0, 1, so 1 for expressed, 0 for not expressed, then I could write the dynamics in some of this form. So at each time step, a gene I will be expressed if this signal it receives exceed a certain threshold up to some noise, okay? Z here is a random variable which mimic biological noise. And I can, so the magnitude of the fluctuation, which is the level of noise in the system, is controlled by a parameter T. Now, neural networks have been modeled for many years since Rosenblatt by pretty much in the same way. Here, the convention is normally to use neurons as Ising variable, plus or minus 1. So there is a sign function rather than a step function. But it's basically expressed in a similar form. And it is well known since Hopfield model that one can store P patterns in this network. Think, for example, a visual pattern. We can represent it as a binary vector, a vector of n entries, each one for each pixel, which takes value plus or minus 1, depending on whether the pixel is blank or white. And then Hopfield suggested that if we choose the couplings between neurons in this form, then this pattern will be attractor for the neural network dynamics, meaning that if we initialize the network in some configuration, as time elapses, it will reach one attractor, meaning that it will retrieve one memory. Okay. And so we thought we could do something similar with genes, so encode cellular attractor in couplings between genes. And we started thinking, what kind of attractors are cellular attractors? So they are, first off, hierarchically organized. And this is because of the cell differentiation cascade. So two cells which differentiate quite early are far away, whereas two cells that differentiate quite late are closer. And also they are dynamical entities because they cycle through the cell cycle. So there is a resting phase, synthesis where duplication of DNA occurs, and then an interphase, and mitosis where the cell divides. So the idea is basically to choose the couplings between genes in such a way to encode a hierarchy of cycles. Okay? And to illustrate ideas, we stick to a two-level hierarchy. So we have a stem cell at the top, daughter cell at the bottom, but each of these guys, not a fixed point, is a cycle. Okay, so rho is an index which vary and denote the stage of the cell cycle, whereas the index mu denote just the cell type in this differentiated layer. So what are the patterns now? Well, are basically vectors of 1 and 0, which tell us for each cell type mu and for each stage of the cell cycle rho, which gene is expressed and which is not expressed. Okay, then we took inspiration from neural network technologies from the 80s. So after Hopfield influential work, a few years later, Sopolinsky-Cantor showed that it's possible to store a sequence of patterns if one applies a simple shift of index to the Hopfield recipe. And then if one loops this sequence, one has a cycle, okay? One can store a cycle. And the other thing that people did about in the same years was to consider, was to embed in networks a hierarchy of patterns. In this case, patterns are fixed point. And they consider patterns which are generated by a Markov process from layer to layer, okay? At the initial, in those days, this Markov process was taken as a martingale, but in principle, this is not necessary. And they showed that, that for that type of process, you could choose couplings between neurons in such a way to embed these patterns. So basically, what we did was just to combine, if you like, these two recipes, cycles and hierarchy, adapt from the Ising spin to Boolean variable, to 0-1 variable, and we did this for a more general Markov process. In particular, we had a conjecture that there might be stages in the cell cycle where two different cells are most similar to each other. And this is because, for example, in the mitosis phase or the synthesis, there is such a huge machinery deployed when the cell divides that it's quite unlikely that this has been reinvented for each cell type. Okay, so this was the idea. And therefore, we considered a Markov process which displayed this phenomenology. There are phases where cells are closer together, and they are also closer to the stem cell. Okay, so this was the model. For simplicity, we just consider a three-stage cell cycle with a single phase more similar, which we call phase 2. And then we use standard statistical mechanics to go from the dynamics of microscopic variable, which is the gene expression, to a set of microscopic variables, which are basically the correlation between the system configuration and the pattern that we are going to store. And in the thermodynamic limit and to infinity, so large number of genes, you can get a set of closed equations. And here I'm showing the results. So here, the full line, M row, represent the correlation of the system state with the stem cell pattern. And the dashed line, the correlation with the daughter cell pattern. And what we have is that if the noise level is very high, then the system doesn't show any correlation with any pattern, okay? Everything just fluctuates stochastically, and we do not have a retrieval, so to speak, of anything. Then there is a critical value of this noise where the system shows high correlation with the stem cell, okay? There is also a residual correlation with the daughter cell, but this is just because the stem and daughter cell are not independent from each other. They've been generated by a Markov process. And then there is a second transition where the system shows high similarity with a daughter cell, meaning that it has retrieved that cell. And again, there is some resemblance with the stem cell as well. Again, because of correlation existing between the two. So there are three curves, one for each stage of the cell cycle, okay? And then in this phase, basically two of them collapse onto each other, and there is a single one, which is the phase row 2, the more similar, which is more similar to the stem cell. So basically, if we are in this regime, so we place ourselves just above this transition here, And we initialize the system in a daughter cell. So the correlation with the daughter cell is maximal 1. Then as time elapses, the correlation with the daughter cell will go down, and the correlation with the stem cell will increase, meaning that the system just abandons the daughter state and gets into the stem cell state. So what was interesting for us is to see that it takes several time steps for this to occur. Now each time step here is a stage of the cell cycle. So 30 time steps correspond about to 10 cell cycle, which is similar to what they see in experiment. It takes several generations to de-differentiate from a daughter cell to a stem cell. However, this de-differentiation is just driven by noise here, by T. So we wouldn't know how to do this in an experiment, say. So next, we considered transitions which are induced by direct perturbation of genes. So what we do is to perturb a fraction Q of genes in such a way to drive a transition from the daughter cell to the mother cell. So in jargon, we apply a field, a local field, to a number of genes which are aligned with the stem cell pattern, okay? And what we do is the following. So we initialize the system state in the daughter cell. We perturb an increase in number of genes, and nothing happens until we reach a critical value, after which the system transitions from the daughter cell to the mother cell, to the stem cell. Okay? And okay, this critical fraction, it's basically, yeah, between 0.1, 0.2. It depends a bit on the noise level, because if the noise is higher, these cell states are a bit shallow, okay, and it's easier to get out of them. And also it depends on the actual stage on which you apply the perturbation, okay? But it's more or less in this range. So how many genes would we need to perturb? Well, if we include in this model just regulatory genes, so those genes that regulate other genes, these are about 2,500. So that fraction corresponds to perturbing 250 to 500 genes. And it turns out that each transcription factor used by Yamanaka is actually involved in regulating many genes, like close to 100. So basically, this was nice because it seems that indeed you would be able to trigger this transition with a few transcription factors of this type, 3 to 5. Okay, so perhaps it's a good time to make a small summary of this first part. So far, we have constructed a model with a hierarchically organized cycles. We have investigated reprogramming within this paradigm. We have seen that it takes several cycles to de-differentiate a cell from daughter to mother. And it can be achieved with a realistic number of transcription factors. And we also seen that the efficiency of this transition depends on the stage of the cell cycle you're at. So this model was nice because it suggested a natural target for this experiment. So what is the main limitation of the model? Is that basically this interaction were chosen ad hoc, so to speak. So just combining neural network technology, okay? So they are not biologically grounded. And the main limitation is that these neural networks are usually formulated in terms of dense connection. So everybody connected with everybody else, whereas gene regulatory network are sparse, okay? So this was the motivation to adopt a second modeling approach based on bipartite graph. And also I wanted transcription factors somehow to be in the game explicitly, okay? So I considered a model with N genes, okay, the blue circle, and P transcription factors, which are assumed to be proportional to N. And we have an error from I to mu if gene I code for a protein that makes part of transcription factor mu. And an error from mu to j if transcription factor mu regulates the expression of gene j. And this regulation can be excitatory, inhibitory, or absent. Okay, so this is a bipartite graph, which is sparse because lots of links are 0 here. And of course, you can characterize its structure in terms of in and out degree and of transcription factor and gene, okay? And then you can consider some dynamics on it, okay? So you can model gene expression dynamics similarly to before. So you say that gene I will be expressed if it receives enough regulatory signal from transcription factors. And then you need an equation for transcription factor, okay? So this tau mu, we loosely call them transcription factor concentration. So what would be a sensible dynamics? Well, of course, it depends on our choice. We thought that a sensible choice was to assume that transcription factor execute a end logic, meaning if a transcription factor is a protein complex, then to be synthesized, all the constitutive protein should be there, okay? So all the genes that express those proteins should be on. So this was a choice. Then we also considered a second version with the idea that sometimes the nature finds alternative pathways. So maybe even if not all the proteins are around, but a few are around, maybe some regulatory effects still follow, okay? So we refer to this as the OR logic. And in this case, we take the transcription factor concentration to be equal to the number of active genes among those that contribute to transcription factor mu, divided the total number of genes that contribute to transcription factor mu. Okay, so this is what you would do, for example, in model of epidemics or contagion. Okay, you basically need at least one node to get a non-zero probability to get infected. Okay, so now we substitute this inside the gene dynamics, and we get the following. So for the OR logic, we then get a linear threshold model where there are here effective pairwise interaction, Jij, which come from the fact that a gene J contributes to a transcription factor mu, which regulates a gene I. So this creates an effective interaction J to I. And so this interaction may be reminiscent of neural networks interaction of the Hopfield type. Although here the psi are not just plus minus 1, but can also be 0. So these are a so-called Hopfield model with diluted pattern. And I studied this in the past. They are quite nice because the dilution in the pattern switch the retrieval from sequential to parallel. So this model, a neural network that can retrieve an extensive number of patterns at the same time. However, here the situation is pretty different because we have psi eta rather than psi psi. And this is a rather crucial difference, okay? They even take values in different sets, and this create a symmetry in these links, okay? So you can't do any of this equilibrium analysis that we did before. You really need to solve the dynamics to understand where the system is heading to, what are the attractors, what are the patterns. And with the end logic, we are in a similar situation. So we, again, have a non-linear threshold model now, again, with asymmetric and sparse, but this time a multi-node interaction rather than pairwise. Okay, so again, what are the steady states of this? Okay, so before solving the dynamics, we asked a very basic question, which is, for which values of the parameters, of the model parameter, would this system allow non-trivial attractor in the first place, okay? And the answer is that, well, each finite system in the presence of noise is ergodic, okay? Because if the system is finite and everything can fluctuate due to the noise, then I can reach every configuration from anything else. So this would only have a trivial attractor. So for a system to sustain a multiplicity of non-trivial attractor, then I would need to have an infinite system. So a system residing on the giant component of a regulatory network. So the question boils down to understand under which condition would a giant component emerge in the network, which can be investigated by percolation theory. So this has been done before for the OR logic. So we adapted the theory to the AND logic for a bipartite directed graph. And what we find is the following. So for a giant component to exist, you find a condition which tells you that the average in degree of transcription factor has to be sufficiently small. So this is just the probability that a gene is in degree 1. Okay, it doesn't really matter. So when we think about this, the in degree of a transcription factor is the number of proteins that contribute to the transcription factor. So if you like, it's the size of that complex. So you need the transcription factor are relatively small complexes. And also what we see is that this system have a bistability. So for the same range of parameters, you may or may not have a giant component. For the giant component to be the only stable solution, then another condition needs to be satisfied, which basically tells that the average out degree of transcription factor has to be sufficiently large. And what is the average out degree? The out degree is the number of genes which are regulated by a transcription factor. So transcription factor should regulate sufficiently many genes, okay? And talking to colleagues in biology, they said that it is indeed close to biological understanding that transcription factors should be relatively small complexes which regulate many genes. Okay? So this was a nice picture of this model. And then after this, we solved the dynamics of this model. So I will illustrate this for the OR logic first. So as a reminder, we have a gene expression dynamics like this, where for the OR logic, this signal that the gene receives is just this quantity. So here we have all the neighboring gene and J that interact with I, okay? So we are calling NDI just the states of the in neighbor of gene I. And Z was a random variable for biological noise. This is just drawn from some distribution whose cumulative we call phi, okay? So we are interested in quantity like the activation probability, meaning the probability that a gene I is expressed at any time. So what is this? So if I look at this equation here, I have that the probability that Ni is 1 is equal to the probability that Z is less than Hi minus theta I, which will happen with a probability phi t, okay? And then I do not know the state of the neighbors of I, so I need to average over them, okay? And this averaging is difficult. The main reason is that it's not a closed equation. So here you have a marginal for single site, and you have to average on a probability, a joint distribution of multi-node. However, there is a powerful method introduced by Mézard and Paris in 2001, called the cavity method, and this is exact on beta lattice, that is an infinite tree. And the main idea is that if you are on a tree, if you remove a node, I, then the neighbors are just independent of each other because there are no loops. And therefore, this joint probability of the neighbors just factorizes in the so-called cavity graph, where I have removed the node I and all the links, okay? So basically, the method consists in writing this equation first on the cavity graph, so that you have a closed equation for these cavity probabilities. And then once you have this, you can get the marginal probabilities in the real graph. Okay, in dynamics, you can do something similar. The situation is a bit more complicated. However, it gets very easy again in the so-called fully asymmetric network. That is, those networks where all the links are unidirectional. So if J is an in neighbor of I, then it doesn't receive a signal from I. So in this graph, then it's true that it's still true that if at any time I eliminate a node I, the probability of the neighbors of I factorize. And also this probability in the cavity graph is the same as the probability in the real graph. Why is that? Because if I eliminate node I, there is no way in which node J can ever realize that, because all these nodes are unidirectional, so it will never receive feedback. So basically, if I insert now this expression in here, I have then closed set of equations for these marginal probabilities, okay? So I basically need to average this function over the neighbors of I, but the neighbors of I are distributed in a factorized form, which can be expressed in terms of the activation probabilities of the neighbors themselves. Okay, so in principle, I can just run this dynamics from any initial condition. I just iterate and until convergence. Now, these steps can still be tricky because for each node I and for each time step t, we still need to average over all the configurations of the neighbors of I. Now, each neighbor can be in two states, 0, 1. So the number of configuration of the neighbors is 2 to the neighborhood of I. Okay, so there is still a exponential complexity in the degree of nodes I. And this in power law network is a real bottleneck. Okay, so in power law networks, so you have nodes with lots of neighbors, and still it's a computationally demanding task to do that. So what we did is we showed in recent work that this complexity can actually be drastically reduced from exponential to polynomial for graph where the interactions are binary, so plus or minus j, by dynamic programming techniques. So I don't have the time to fully explain the method. The details are in this paper, and the code is on GitHub. But basically, the idea is that if your interactions are binary plus or minus j, out of all the configurations of the neighbors, there is a much lower number of field of signal that they can realize, okay? And these signals can actually be computed very efficiently recursively. So, yeah, with this computational speed up, you can solve all trajectories for activations. So you can indeed get the activation probability for each node versus time, as we did here. So this is time. And these are the nodes I. They are actually ranked according to the final value, the value in the final state. So that's why you have this nice color pattern. And of course, once the system is has converged, you can look at the steady state, okay? For example, at the distribution of these activation probabilities. Okay, the method works well. So this result from the cavity method on top of Monte Carlo simulation. the big advantage of using cavity is that this distribution are really spiky, so you need a lot of resolution to resolve these peaks. And of course, getting that resolution in Monte Carlo needs you have to run a very long trajectory, which for this network size takes several days on a GPU. However, dynamic cavity with this computational speed up just converges in under a minute. And yeah, and I want to stress that, so in this network, for example, we have considered that the largest degree was 800. So you couldn't do that without this computational speed up. Okay, so this in particular allows us to resolve the distribution of activation probability in the steady state for any value of the control parameter. So these are just 3 snapshots at 3 different values of the noise. So you can see, for example, low values of noise, lots of genes are frozen in state either 0, 1. So the activation probability are also frozen in 0, 1, and you have these 2 peaks. And then as you increase the noise, genes fluctuate stochastically, and therefore the activation probability will be roughly half. And therefore you see that as increasing the temperature, you get these peaks traveling towards the center. Yeah, we can, so from a general point of view, you can see these distributions are really heterogeneous, okay? They are multinodal. So indicating indeed that there are stronger heterogeneities in the network, and you wouldn't be able to get this, for example, from mean field method, okay? So cavity method is really nice because it pins down to the individual node in the network. We can rationalize many of these peaks through a simple argument. I will not enter that. And we can, in principle, go beyond the activation probability. For example, we can compute joint activation probabilities, so correlation. You can adapt the cavity method to actually compute also those quantities. Okay, so far so good. However, in this network, we have considered just fully asymmetric links, okay? So this network would only have one attractor, and information just flow and stabilize somewhere. So, for example, this network wouldn't be able to sustain a different cellular attractor, okay? So to have a multiplicity of cellular attractor, you need to introduce a degree of symmetry. And this is what we did next. So basically, we introduced some finite probability that transcription factor mu regulated gene I, given that gene I contribute to mu itself. So it's basically a kind of self-regulation, okay? Gene I contribute to a transcription factor that regulated I itself. And in this case, we do see a multiplicity of attractor at very low temperature, at very low values of the noise. And then we looked at how similar these attractors are. So what we do is to run many different dynamics from different initial conditions. You wait until the steady state is reached, and then you look at how similar different steady states are. And this is the distribution of similarities. So the left panel is for the AND logic, and the right panel for the OR logic. And what you see is that if P is 0, so there is no self-interaction, then there is only one attractor, so all the dynamics converge to the same attractor, so the similarity between different dynamics is just 1, is maximal. And you have a single peak in 1. Upon increasing P, this symmetry parameter, then you have that the different run converge to different attractor, and quite interestingly, they have a typical similarity between them, which is basically the node of this distribution. And an interesting aspect is that this similarity seems to be lower for the AND logic than for the OR logic. So basically, here are two take-home messages. One is that self-interaction seems to be crucial, at least within this model, to get a multiplicity of attractors. And this cooperative effect of the AND logic, so many genes need to collectively regulate others, seems to promote diversity of attractors. And again, both these features seems to be really common in gene regulatory networks, so they might be there for a good reason, okay, for sustaining multicellular life, the possibility that the same network sustain different attractors. Okay, so the summary of the second part, yeah, we have first used percolation theory, and from this, we learned that for a cellular attractor to exist, we would need transcription factors to be typically small complexes which regulate many genes. And then we have solved the dynamics of such systems. We have introduced dynamic programming algorithm to calculate a gene expression profile. This can be, in principle, compared with what we get from experiment, from mRNA counts. So this is quite nice. We saw that this distribution have a very rich structure. Some of the features can be rationalized. And then we extended this to include the bidirectional links. And we saw that when we do so, a multiplicity of attractors emerge at low temperature, at low noise. And we have seen that multi-node interaction seems to favor diversity of attractors. So, okay, there are still many unanswered questions still many unanswered questions, many of them pointing towards information processing capabilities for sparse system, other pointing towards a multi-node interaction. I hope to discuss more with you about this. And let me just acknowledge my great PhD students, Ryan Hannan, Giuseppe Torrisi, and Christian Harry. They were all funded by EPSSC. And a list of colleagues who have, to different extent, contributed to either to this work or to closely related idea. In particular, Reimer Kuhn, who has co-supervised with me, Ryan and Giuseppe. And yes, that's it. So these are the references. Many thanks for listening.
Any questions or comments?
Thank you for the nice talk. In order to, one of the ingredients in both models you have shown is the stored patterns, the stored memories. How do you choose them randomly with some biological idea or just
yes,
at random?
So for the previous model, the neural network, in principle, you can plug in the gene expression profile of data that you get from experiment. So the PhD student actually did this. Of course, we do not, especially at the time, we didn't have too much fine details about gene expression at different stages of the cell cycle. So more usually, you just get gene expression in some cell type, which is probably some average across the cell type. But if you had this information, in principle, you can just put those into your recipe for the couplings. In the second model, things are more complicated because this network between gene and transcription factor, nobody really knows. Especially when coming to cooperative effect, so everybody knows that they are present, but lots of data are actually for single protein transcription factor. So you know a gene expressed for some protein, and you know that this protein can act as a transcription factor for another, but nobody really knows how different proteins act together. So there is some evidence, I mean, lots of evidence for that, but the information is still a bit patchy. So I think from the data side, we are not really at the point to get enough information to calibrate this model, but perhaps you could hope to do the other way around. You have a protocol to, given the interaction, to know the gene expression profile and also the correlation. And then maybe you can try to infer some of these interactions. But the situation on data side is getting much better every year.
What do you put in your model? Random or what?
In the model, random patterns. So in the first model, as I said, also data, actual data. Some studies, so just to add a comment, the network statistics was indeed from gene regulatory networks. So for example, the statistics of degrees, all those things, we did take from the gene data set, yes.
In the kind of bipartite network that you build, I mean, in which you have the genes, I mean, interact with the transition factors, I mean, because, I mean, all those genes who, I mean, build proteins that make the complex. I want to comment that in many cases, so in a way, this is assuming hetero complexes. In many cases, I don't know if you have heard from biologists, in many cases, they are homodimers, trimers. So the first gene, so it's a complex, but it's, I mean, dimers, trimers, tetramers, whatever, from the same protein. So codified by, for instance, just one gene.
Yeah.
This has a strong kinetic effect. So you go to a Hill equation and it's the Hill exponent and so on. I wonder whether, I mean, this influences your study. I mean, in case you have one gene, but codifying a complex with several monomers, let's say, that are
this is really interesting. So at the moment, in the multi-node interaction, you just have a product of things. You don't, but one can use as a power, okay? And it would be interesting to consider, yes. Mm-hmm.
Well, then let's thank Alessia again.
Yeah. Ok, piacere Alessia.